Okay, sorry about that. I hope it doesn't happen again. Welcome and thanks for coming. It's a real pleasure, and it's not only a sentence of being a pleasure, it's a real pleasure to introduce our speaker of today, which is Professor Jorge Viñals, which is Professor of Physics and Astronomy at the University of Minnesota, and the Director of Graduate Studies. Jorge got his PhD in Barcelona back in 1983, although part of his PhD thesis was in fact done here on the campus of the University of the Balearic Islands. From there he moved as a postdoc to Temple University and in Philadelphia and Carnegie Mellon in Pittsburgh. And from there, in 1989, he got a position in the Supercomputer Research Institute at the Florida State University in Tallahassee, where he stayed until he moved in 2004, from 2004 to 2010, as director of the Institute of Advanced Materials in McGill University in Montreal. And finally, in 2010, he moved to University of Minnesota. In 2010-2015, he was the director there of the Minnesota Supercomputing Institute. Jorge has been on his research side. He's working on a number of topics, statistical physics, kinetics of phase transitions, nonlinear physics, and pattern formation in always a high degree of numerical techniques. I'm happy to say that on the kinetics of phase transitions, back in 1985 we had 2 joint papers, one on the Carroll normalization group, and in the period of 91-93 we have 3 joint papers with Emilio, Raul, myself and Jorge on pattern formation. He's presently involved in a number of topics on liquid crystal dynamics, suspensions in nematics, defects in nematics, and I think this is what he's going to tell us about today. Thanks for coming, Jorge.
Yep. Estic molt content d'estar aquí, no per portar-te la contrària. Tot i que soc foraster, i ben foraster, vinc de bastant lluny, però cada vegada vinc aquí em trobo com si estigués a casa meva. Gràcies a tots per venir i per tenir-me aquí un parell de dies. No sé, seguiré en anglès perquè m'aturaré menys, però potser m'animi i... Molt bé. El que digui el jefe. Us parlaré d'una recerca que he fet els de... I'm going to tell you about some research in liquid crystal-based suspensions. The research has been done with my student, Chris Conklin, who just graduated last November. And it's been done in close collaboration with experimentalists, and everything I'll be showing is really a mixture of experiments and theory. And the experimentalists belong to the group of Oleg Larentovich, who's at the Liquid Crystal Institute in Kent, and 2 of his students, Shenghui Peng and Satya Paladugu. So first, what's a nematic suspension? A nematic suspension is a suspension, that is, you place solid inert particles in a fluid, except the fluid is not a normal fluid, but it's a liquid crystal. And not only is a liquid crystal, so you set up the experiment so that the liquid crystal is in the nematic phase. A liquid crystal is a system of ellipsoidal or cylindrical particles that are actually a liquid. So if you imagine the system at very high temperatures, particles flow freely, the orientation of these little cylinders is random, and it behaves for all practical purposes like a normal isotropic fluid. When you lower the temperature, these molecules tend to align. So there is a state of long-range orientational order in which all the molecules point on average in the same direction, but they still flow. It's still a fluid. It has the regular properties of a fluid, but it has a bit of a liquid or elastic light response, because now you can shear it and play against the orientation. These suspensions were introduced, as far as I know, 10 years ago, and the reason, which I'm not going to talk about today, is to eliminate sedimentation. When you have a suspension, particles eventually, because of the force of gravity, they move to the bottom. The thought was that if you have a nematic fluid with long-range orientation, so I'll explain this a bit more later, but just to introduce the subject. These lines denote the orientation of the nematic molecules, these little cylinders in the fluid. If you place a particle inside that fluid, the thinking is, and I'll be more precise later, the thinking is that it will distort those lines, so they will not longer be able to be oriented uniformly. This distortion, if done right, can in fact cancel the force of gravity, and therefore it is possible under the right conditions to have particles levitating forever. That was the thought of doing the experiment, and this was just a demonstration. This is a particle, and this is through microscopy. It's a very easy experiment because these particles are micron size, so you can see them. It has lots of unusual properties which I will not discover, but this is just one. The particle, of course, levitates, doesn't touch the bottom, but the distance at which the particle settles at long times actually depends on the system size, because now the liquid is ordered. In order to place the particle, you have to distort the orientation of the nematic, and therefore where the particle sits now depends on the size of the system, because that determines also the orientation of the nematic. So it has, they discovered a number of unusual properties which I will not discuss today, but for the purposes of the talk, think of a liquid crystal in a nematic phase, in some cases with, and in some cases without particles. And what I'm going to focus is in a slightly different problem, which is not only the particle distorts the nematic, so it can give rise to elastic interactions with the boundaries or other particles, but there's an issue about topology, and that's the one I really want to focus today. So imagine you have a particle, a spherical particle, in a nematic. So let me imagine that at some place in infinity, all the nematic molecules are pointing in the same direction. They're oriented somehow by the boundaries. However, there's some chemistry, some electrostatics between the particle and the nematic. So the nematic is not, of course, invisible to the particle. Due to whatever reason it may be, the orientation of the nematic molecules relative to the particle is fixed, again, by chemistry and electrostatics. So they could choose to orient perpendicular, parallel, or at some intermediate angle, whatever it is. So for the sake of an argument, let me imagine that these nematic molecules, these cylinders, like to orient perpendicular to this immersed particle. And this is what's shown here schematically. If this is the case, then if you now trace a circle around the particle and you integrate the angle variation as you go along the circle, you do a closed integral, and it doesn't integrate to 0. It integrates to 2 pi in this diagram shown. That's a topological defect of charge 1. The circulation of the angle is 2 pi. So as far as the nematic is concerned, having introduced the particle has in fact created a topological defect of charge, in this case, +1. Again, you just go around the particle, you integrate d theta, and the closed integral over a closed contour of d theta is not 0, it's 2 pi because of the defect. Now, you have a situation in which the particle has created a defect. Far away from the particle, the charge is 0. If you do the same exercise and you integrate the angle through the boundary, you just get 0. The angle doesn't change, so a contour integral gives 0. And because charge has to be conserved, then that means that somewhere in the fluid, a -1 defect has to nucleate. So every time you put a particle, the particle is a defect, and to conserve charge, plum, a defect has to nucleate somewhere or more, but they have to be such that the charge outside will be 0. So this is the problem I want to discuss today, to understand the states and the transport in systems that have pairs of particles and topological defects. What happens? Well, this is a bit of a, there's lots of possibilities, but a bit of things I'll discuss today. So first, let me imagine that the particle is very small or very large. Let's start with very small. There is another characteristic length in the nematic, which is the ratio between the elastic constant, how much it refuses to be distorted, K, versus W, which is the anchoring energy, how much energy is involved into fixing a particular orientation. So how much, if the anchoring is very strong, that energy is large, and the particles are fixed. If the energy cost to misorient it is not very large, then that's given by this quantity W. So the particle that you immerse is very small compared to this length. The nematic is very rigid compared to the anchoring force. The nematic basically ignores the particle. Just the nematic stays straight and really ignores the particle. As you make the particle bigger and bigger, then you start forming anchoring perpendicular, something which I sketch here. And in 3 dimensions, in the case of a +1 particle, you really have a few choices, but here are 2 of them. One of them is called a Saturn ring, in which, and I'll give you more details later, but just to motivate what I'm doing, a Saturn ring is a -1/2 disclination. I'll tell you later better what this is, but this defect right here is a -1/2 defect, which actually wraps around the entire particle. That's why it's called a Saturn ring. And the global charge of this ring is -1, and that cancels the +1. So this is a possible configuration that the system would adopt if you immerse a particle. This actually happens to be the case if the particle is large, but not very large. As the particle gets larger, then the situation that typically appears is this, in which this point here is a point defect as opposed to a line. This is called a hyperbolic hedgehog. It's a -1 defect, and I'll give you more details later, but this is how the configuration actually looks like, and again, it's rotationally symmetric. So by introducing the particle in this nematic suspension, either nothing happens or you form this particular defect. This is an experimental image through polarized microscopy. Here you see the defect, and this little smudge here is the hyperbolic hedgehog. That's what you would see if you do the experiment. So you eliminate the sample from the bottom, you see from the top, and then you put it through cross polarizers to see the distortion to the light propagation that's caused by the defects. And this would be a Saturn ring, what you would see in an experiment. This is 2-dimensional, so I have a bad 3-dimensional visualization capability, so this is how it actually looks like in 3 dimensions. It's a point defect, and these would be the lines parallel to the direction of the nematic locally. Well, what is this good for? A number of things have been done in the last few years, and I'll touch upon a few of those. The first one is that if you now remove particles, but you fake the defect somehow, if you manage to create a nematic pattern which is distorted similarly to the way it would be in a particle. So here I'm showing really a +1 and a -1. If you manage to have a nematic configuration such as this, then an electric field is capable of producing charge separation, and I'll explain how that happens. Once charge is being separated, then it produces systematic flow. So you can actually use a particular directional configuration with or without particles to create average velocity fields that don't average to 0 with an applied electric field that could average to 0. This is called electroosmosis, by analogy with the electroosmosis in electrolytes. You apply a field and there's fluid motion. You can also induce electrophoresis. So if now you place the particle again in this nematic medium and you apply a field, the particle and the defect manage to create a semi-complex flow such that then the particle displaces. So you can actually move particles in controlled directions and with controlled speed by imposing an electric field through this charge separation, which I'll explain. More exotic perhaps, one can tailor the nematic pattern to create chaotic mixing at the microscale, something which is difficult to do in a scale of microns for a fluid of this viscosity. More recently, people have used that to guide by bacterial motion. So here is the pattern of the nematic that's imposed, and what you see here are actually some type of bacteria that swim in circles. And just to mention across the pond, in Barcelona, there's a group that's actually using the same strategy to induce clustering and guided motion of ensemble of bacteria in a nematic matrix just by shining a laser on the system. So there are a number of applications, the goal of which mostly is to be able to control flows on demand, to be able to control particle motion on demand, and with applications in microfluidics, in guiding light has been used, and also in biology. You can use it for cell sorting, for separations, and various other phenomena. So let me start from the beginning. This is a sketch of a liquid crystal molecule, and this would be a nematic phase in which all the molecules on average point in the same direction. The order in the nematic is described in most of what I'll talk about with the so-called director, which is the local orientation in an element of volume. It's a unit vector, which just tells you in which direction the molecules are pointing. The elasticity of the phase is given by some excess free energy when there is a spatial distortion of the order parameter. It has terms that look like this. I'm not going to talk much about them, but this is well known. And it describes the various ways in which you can distort a reference ground state by splitting the configuration, by bending them, or by twisting. What I'm going to do is study flow, but the flow is simple. It's low Reynolds number, so it'll be just Stokes flows. It has the other complication that the viscosities are anisotropic because the phase is no longer isotropic, but that doesn't play much of a role, so I will not be talking a lot about that. There are two things that actually control the physics of what I'm going to tell you. One is that in nematics, even though you may or may not try to purify them, they always have some embedded ionic species in them. Your liquid displays on TVs or computers try to be as pure as they can, but they always have embedded ionic particles, and they will be an integral part of the story I'll tell you in a minute. So even if you try to remove them, you can't. They survive the processing, or they come from electrodes or from the boundaries of the cell. So I'm going to, the physics is based on two properties. One is that the molecule, the liquid crystal molecule itself is dielectric, but it has anisotropic dielectric properties. So there is a dielectric constant parallel. It's not written here. It's a dielectric constant perpendicular, and then delta epsilon is parallel minus perpendicular. So there's a polarizability along the axis of the molecule, different from a polarizability in the perpendicular direction. So this is one anisotropy which matters. Another anisotropy that matters is the ionic conductivity. These ions move in response to perhaps electric fields, and the mobility of these ions is different if it's perpendicular to the local nematic orientation, or delta mu is parallel minus perpendicular. It's different if it's along the parallel direction. So typically ions will have an easier time moving parallel to the local direction of the nematic rather than the perpendicular direction. So this is how the first set of experiments, which I'm going to describe, is organized in order to understand a little bit the physics, because it's not that the physics is very complicated. The equations are well known. This has all been well known for a long time. But because there are several equations and several phenomena, it's hard to know which one is operating in any given experimental condition. So the first things that we've done is to study experimentally and theoretically and computationally configurations in which the order parameter, the nematic director, is fixed. So imagine you have an experimental cell such as this. This would be the top, this would be the bottom, and there's a nematic liquid crystal thin film in between the two layers. And what they do is, through photo patterning, they can impose the orientation of the nematic on both surfaces. And if this is thin enough, then this ends up being the orientation of the nematic inside the cell. So it's a nematic with a fixed, externally fixed director orientation. So this could be one example. Again, you do this by photo patterning, and that's the orientation of the nematic that you would inherit. The purpose of doing this, as I'll show you in a minute, is try to understand what actually goes on here. And so this is it. When you have a normal electrolyte, the first thing that you have to do if you want to induce motion is to create charge separation. The electrolyte is neutral. If you apply a field, nothing happens. But if you have means by which positive and negative charges separate spatially, then you can create forces and you can create motion. In the typical case of electrolytes, you need solid surfaces, immersed particles or boundaries to do this. Here the mechanism is slightly different, and let me illustrate this with this slide. Imagine, as a physicist, you start with a periodic case. So imagine that you photo pattern or you create a configuration with the nematic order is periodic. So you're looking at from the top, this would be a quasi two-dimensional thin layer, and from the top, this would be the pattern of the nematic, just periodic. Now imagine that you apply an electric field from left to right. This liquid has some ionic impurities, as I said, and imagine which is typically the case, that the conductivity parallel to the nematic director is bigger than the perpendicular conductivity. So when you apply the field, the positive charges will try to migrate to the right, but they will actually predominantly follow the nematic lines, and in doing so they will congregate in the middle, whereas the negative particles will try to move, this is initially neutral, the negative particles will try to move opposite to the direction of the field, which means as they move, they will also follow the nematic lines, and they will congregate along the regions in blue. So just by inducing a field without any solid boundaries, just by the fact that the fluid is anisotropic, you create some charge separation. The positive charges accumulate here, the negative charges accumulate there. Of course, because the field is on, now there is a body force in the positive region that points to the right, and there's a body force in the negative region that points to the left. That's a second order effect now. If you actually reverse the field, this will change. This will be the accumulation of negative charges, and this will be the accumulation of positive charges. But because you reverse the field, actually the force is in the same direction. It's the product of the charge separation times the field. So what happens is, even if the field is oscillatory and the charge separation oscillates with the field, the body force on the fluid is actually always in the same direction. So in this particular configuration, you're going to have a set of a cellular flow in which this region of the fluid will move right and the other fluid will move left. So without any particular boundaries, you have charge separation in the bulk and you have motion. So this is just to explain the mechanism a little bit more. Imagine I write the equation of mass conservation for the ionic species, and this is more or less it. It has a left-hand side, which is a time derivative. Then it has the divergence of a flux. The flux has a number of terms. It has a diffusion term. I'm just going to write it for this configuration, which is uniform in the x direction, and there's only change in the y direction, just for simplicity. So the equation of transport will have diffusion, but there's no change along the x direction, so you'll only have gradients along the vertical direction. There is a driving term, which is E sub x, and calling x the horizontal direction. This is the field that's actually pushing, but because the conductivity now has a tensor, has a parallel and a perpendicular part, there's a yx component that actually creates a flow in the transverse direction. That's what creates charge separation. And then as the charges separate, of course, they create an electric field in the same direction, which itself induces another flux, which is given by a different conductivity. So there's a driving term induced by the field, and there are three terms that can saturate growth. One due to diffusion, which is typically negligible except in regions with large gradients, and I'll discuss those separately. There's a saturation because as the charges separate, they themselves create an electric field that opposes separation. But the two effects now have different components of the mobility or conductivity tensor. And then there's also saturation by oscillation. If you change the field too quickly, you don't give enough time for these particles to separate. In practice, in the experiments, there are various times. The time that matters is the separation time that's due to saturation because of the transverse field, which is relatively small. Diffusion for the periodic configuration is relatively large. So basically what determines the configuration is the balance of this driving term and this saturation term. And you go and do the experiments, and you're able to predict fairly well the experimental measured velocities and the velocities that you would obtain just from basically scaling analysis. And the agreement is actually better than it looks because these computations assume no-slip boundary conditions on the endpoints, and the experiments are instead an open cell, so the velocity is a little bigger. But anyway, the message at the moment is that you can actually induce transport in a nematic liquid by relying on the anisotropy of the mobility. You can also make the same argument with the anisotropy of the dielectric permittivity, but it's a bit more complicated and I will not do it. But relying on the anisotropy of the fluid, you can actually separate phase spatially, charge spatially, and use that to create motion. Yeah?
Yeah, the anisotropy is induced separately.
The anisotropy is just a physical parameter. The dielectric anisotropy is a property of the molecule, and the conductivity is a property of, again, how the species will interact with the medium. It's just the usual mobility.
Can you apply a pattern that is to get the charge separation?
No, you're not, no. Oh, sorry. In this case, yeah, the pattern has been applied.
The surface,
Yes. Yeah, this is an applied pattern like this. Yes. Yeah, if not, nothing separates.
You apply
Yes.
that and then you get
The ions move. And what is the liquid crystal? Is it fixed still? Even for mass force?
No, the liquid moves. The whole liquid moves. The liquid crystal does move. So when I show the velocity, what I mean is the velocity of the liquid crystal.
And why not?
Well, because you have an element of volume and everything is interacting in the element of volume, so what you have is advection of the whole element of volume, which is mostly liquid crystal. It's just that the body force only acts on the ions.
But you're assuming that there is no deformation of the pattern.
So we've done that. Yes, there is some deformation of the liquid crystal due to motion, and that scales with something that's known as the Ericsson number. The experiments are typically in small Ericsson numbers, so distortion is small. There is some, but small. Okay, so once you understand how to do a periodic pattern, the next thing you have to learn in order to control flows is to understand how topological defects influence flow, and this is just a basic topology lesson in 30 seconds. Imagine a nematic pattern such as this one or that one. So the lines that you see are the orientation of the nematic. In the case that we're discussing experimentally, you can fix them by photo patterning also. These are topological defects of different charges. The way this is defined, again, is you take a contour, a closed contour, and you integrate as you go over the contour the change of the angle, and you count the total integral. And the integral is always a multiple of 2 pi. In this case, and I should have reminded you that these are polar molecules which are invariant under sign reversal. These are apolar molecules. They don't have head or tails. So this molecule is the same as this one, not my fingers. So in this particular case, when you go around the defect, you get pi. So this is 1/2 of 2 pi. This is the probably more famous or familiar vortex. If you go around this configuration, you get an integral of 2 pi. So this would be a charge of 1. And in this case, if you now go around the contour, you rotate in the opposite way as you go around, and you get an integral of -1/2. So these are a +1/2, a 1, and a -1/2 defect. If you look at the equations back, they're hard to solve, but if you do it perturbatively in delta is the anisotropy of the mobility and the anisotropy of the permittivity, for the range of the parameters in the experiment, that's basically the equation that you have. Phi is the azimuthal angle, phi, as it goes around. R is the radial distance from the defect, so there's a driving term for the charge, rho. The driving term that comes as 1 over r, that comes from the disclination. It has a certain dependence on m, the charge, and the angle. It's proportional to this particular combination of the anisotropies. You can see how it goes. So in the absence of this term, which comes from diffusion, then just gives you how the charge depends both on distance and angle away from the defect. Very close to the defect, this term matters. The constant here is called the Debye length, which is the screening length of the ionic species, which is microscopic. L is some macroscopic size, so this is a very small quantity. So typically, the solution is this equals this, except very close to the defect, where the diffusion actually saturates the value of rho1. So you can compute that perturbatively. And this is how it looks. now perhaps I'll skip the details, but when the field is pointing in this direction and you have a plus 1/2 defect, which the arrows still indicate the nematic, if you have a plus 1 defect and the field is oriented that way, what you have is a concentration of negative charge near the defect. And you can sort of see if the positive charges try to flow this way, they're spread out, so you hardly see them. The negative charges that come from the right end up converging towards the defect, so you get a charge accumulation in the defect. If the charge is plus 1, this accumulation happens on both sides, so what you get is a dipole, a charged dipole. The positive charges accumulate on the left, the negative charges accumulate on the right. If you have a minus 1 defect, minus 1/2, then you get a quadrupole. And that you can predict from this equation. What's interesting then is that, well, first you can solve it numerically and verify your code. This is not so, these are singular solutions, at least for the nematic. So in this particular case, what I'm showing here is the charge. The charge can be computed analytically. This is a calculation, numerical calculation on the right-hand side. The charge accumulation actually goes as 1 over gamma, where gamma is a very small number. So this is a very large number. There's a lot of charge accumulation here that depends on the Debye length, but it's not infinite. And then what I'm showing on the right is the velocity field that's created by a plus 1 disclination or plus 1/2 defect. What you find are two rolls in this particular direction. So again, you pattern the nematic as he was asking, you apply an oscillatory field, and this is the systematic part of the velocity that you induce. You induce two rolls. If you have a plus 1 defect, then the charge distribution is different, and this is the flow that you get. It's quadrupolar. It's different. And again, the charge will oscillate, and it'll average to 0 if you oscillate the field, but the velocity doesn't average to 0. The velocity has that particular pattern. And anyway, minus 1/2. One set of things that you can do very quickly is to engineer flows. If you know what a single defect does, now you can put them together and come up with configurations of interest. So this is a particular example in which there are 3 defects. This is a plus 1. There are 2 minus 1 defects here, so that the sum is 0, and this is the director configuration that you would pattern. In this particular case, the charge that appears is given like this, so you'll recognize the dipole and the quadrupole as I was describing before, and now you have a particular charge distribution which oscillates with time. However, the velocity field now that you create is like this, has 4 lobes. This is our calculation and this is the experiments. And what's interesting about this flow is that it goes in from the top and the bottom and it pushes out from the sides. So if you had a microfluidic cell and you wanted to have a suction that sucks fluid from one direction and moves it in the other direction, you could do that just by patterning this particular thing at a microscale in a particular junction. And this would act like a little engine. It pulls fluid from the sides and it pushes horizontally. Lots of things have been done, and I will not bother you with the details, but you can make them rotate, you can create cellular flows, you can mix them. Once you know how to put together these building blocks, then you can engineer flows just by patterning them on the surface. Something that's somewhat interesting, and I just wanted to mention it, is that particles, when you do classical electrophoresis, you put the field in one direction and the particle moves in that particular direction. Here that's not true. The particle actually, this is a different case, and I'll describe this a little later, but I'll mention it here. Imagine that you have a particle with this particular defect, and now you put a field but in the perpendicular direction. And in the context of what I was saying before, a particle hedgehog pair can be modeled by a +1, -1 defect pair. So if you could pattern it, this would be the analog of having a suspended particle. The +1 gives this particular pattern, the -1 is this particular pattern, and this is actually the velocity field. The velocity field is in fact symmetric vertically, which is in the direction, well, I've rotated things, sorry. So this is the direction of the, I could rotate this, but I couldn't rotate the letters because I don't have these figures. So I have to ask for forgiveness. But in the vertical direction, the field is symmetric, but not in the horizontal, which is the direction of the defect. So you can actually apply a field perpendicular and the pair will actually move in the direction of the defect, perpendicular to the direction of the fluid. So you can have a fluid, it's a fluid, but with anisotropic mobilities. You can control the flow relative to the applied field. So let me move to particles, which is the topic at the beginning. The classical case in which you have an isotropic fluid, if you apply a field, say plus and minus, and you have a particle that's polarizable, then the positive charges accumulate as far as they can from the positive side, the negative charges accumulate on the other side. The electrolyte has mobile charges which screen these ones, so here you have pluses and here you have minuses. And because of the field, and now the charge separation which has been induced by the existence of this boundary, then it will push them out and create flows. That's how you would have electrokinetics in a standard fluid. You need these polarizable surfaces. Here you don't need any polarizable surface. You can have any odd surface. The charge separation actually happens in the medium. So again, if I have a particle immersed in the nematic and I apply the same electric field, then perhaps it goes from the left to the right. The positive charges will accumulate here, the negative charges will accumulate there, not because of directly of the particle, rather because of the distortion in the nematic field that's created by the topology of the configuration. And that's what creates charge separation, and this is what creates motion. I'm going to maybe not dwell too much on the defect, on the details, not on the defects. Oops. So we've done a number of computations and calculations. This case is more complicated. You can have some analytic solutions in 2 dimensions, but nothing that's known in 3 dimensions. This is just a schematic of what now a numerical configuration would look like. I have to say that everything I've said so far has been done with a nematic. It is also possible to describe a nematic fluid with the so-called tensor order parameter, which in some limiting case is just related to the nematic like this. This is important, and I will not get into it. It's important because mathematically and computationally, the configurations that I've been showing you, and I'll show you even more, involve particular singularities of the defect. In the ones I've showed you, the singularities are really imposed by the boundaries. And because of the bi-length, those singularities are regularized in the medium. So it doesn't really matter for the nematic, for the cases in which the nematic order is imposed. In the case of suspended particles, these are truly singular points, and it's difficult to obtain them analytically, of course. There are solutions in 2 dimensions, but there's no known solution to this in 3 dimensions. And it's also difficult to do it computationally because you have to handle singular solutions. So in some cases, people prefer to introduce this alternative representation in which Q now is a tensor, and this tensor actually has no singularities in the defects. It's just eigenvalue crossing that tells you where the defects are. So we've done some calculations here too, but I'll start with this. So this is a situation where you have a particle and a Saturn ring, and this is what you would see if you had an axisymmetric cut. This would be the Saturn ring going around, and this would be the hedgehog. The first problem that happens with this representation, and I don't want to dwell into this too much, but maybe just to tell you, is that this should be a point defect, but it's not a point defect, it's actually a small ring. So if instead of using a singular field, you replace it by a tensor, and then use what's known about the liquid crystal properties and description in terms of this tensor, this defect actually opens up in a ring, which you then computationally have to resolve and deal with. But anyway, this would be the configuration. And you do in fact get charge separation. So this would be the sphere, this would be the defect down here that you don't see. The electric field is pointing up, this is the distribution of charge, and this is the velocity. And you can rationalize this sort of easily in terms of the picture I gave you before. When the field encounters the region without a defect, the charge separation or the charge accumulation is more pronounced than in the region where you have a defect. The defect sort of diffuses the accumulation of charge. So you would expect to have a larger gradient in one side than on the other. The defect breaks the symmetry. And then once you have this symmetry broken, that's what causes the flow around the particle. So this would be now the hydrodynamic of the liquid crystal. So in principle, with that particular thing in mind, you would say, well, the flow is going to be directed towards the defect, because again, the defect minimizes the concentration of charge. So you take the model, you take the physical parameters, you do the calculation, that's what you obtain, except that in the experiments, the flow goes in the opposite direction. It actually flows from the defect upwards, as opposed to flowing downwards. The reason for that is not entirely known, but I'll just tell you a little bit of the things that come into play. So imagine again I have a sphere and I have a hyperbolic hedgehog. In 2 dimensions the solution is known, and one actually can find the solution for the charge in 2 dimensions, and then approximately you can find the solution for the velocity field in 2 dimensions. Then you can ask yourself the same question I asked before, replace the particle hedgehog defect by an equivalent topologically +1 and -1 defect, and try to understand what is the charge that corresponds to this dipole, the topological dipole, and the flow. It turns out that the charge, which I'm not showing for the + and -1 dipole, is the same at long distances and the particle defect pair. They are topologically equivalent, and the charge at long distance are equivalent. However, the flow is completely different. The flow that you get here is from the particle towards the defect. Here, what you get is something that goes in the opposite direction. So one particular, not only the flow is reversed, here you have 1, 2, 3, here you have 6 vortices with a little bit of imagination. This corresponds to this. This is just a dipole of 2 defects in which you just have 2 vortices. Here it circulates this way, here it's somewhat messy. So 2 configurations that would give rise to the same charge at long distances, and they are topologically equivalent, they actually have a very different flow at long distances. That's because, perhaps unsurprisingly, the inner flows also matter. That is not only the long distance driving force from the charges, but also what's the short scale flows around the particles, which are different if you have a particle than if you just have 2 defects. But interestingly, if you just do it in a thin scale in 3 dimensions, what you actually find is that the flow reverses compared with the 2-dimensional case. And not only reverses, but it agrees with the experiments. Somebody, oh my goodness. Sorry. I should have started drinking earlier. I didn't know I was going to get the liter. Yes, getting there. So the message, the summary of the message here is, even though one can make certain arguments on the topology, or based on the topology of the configuration, that seems to be perhaps sufficient to determine the charge separation, but there are qualitative changes in the flow field. You can change the number of rolls and even entirely the circulation. This is a sort of a nuisance because in conjunction to this, which is also reversed relative to the experiments, does mean that both analytically and computationally the structure of the singularity does control the long distance properties of the nematic. So this is an interesting problem that we know some things but not others, and it's one of the things that helps to do with experiments, because if not, you do the wrong thing, I guess. There's something interesting, I'll just mention it, because the driving force depends on this difference, the difference in mobility and the difference in dielectric coefficient. It is possible to engineer flow reversals. That is, you can actually reverse the sign of this difference by changing the composition of the mixture, and then the driving force, which is the right-hand side, would change sign. So you can have, for the same field and frequencies, a charge separation that looks like this if this is negative, or you can have it completely different if it just looks positive. The mobility is very hard to change experimentally, but the dielectric permittivity is very easy to change. You just make a mixture of liquid crystals and you can make this positive or negative. This is something that's also useful for engineering that you cannot do with regular electrolytes, which is just reverse the flow. These are experiments. You take a nematic and you can change the composition of a mixture of two nematics now, so you can actually play with the dielectric anisotropy. The green curve is the anisotropy, so you can make it change as a function of the concentration, the composition of the mixture. The ionic permeability doesn't change very much, but there's a point that they cross, and when they cross, the velocity of the particle switches sign, goes from negative to positive. And you can also do that by changing temperature, as shown here. So there's an additional level of control that's just given by this very simple scaling. Particles interact, and I'll be brief. So once you know what one particle does, of course, you put them together. So these are a pair of two particles with two defects, and you can actually compute the charge separation, and you can compute the flows. What tends to happen in this particular case is that the existence of one particle screens the other particle, so the charge concentration here is higher than on this particular side. And even though the pair translates as if it were one particle, because of this extra screening, the effective interaction between the particles is attractive. So if you have two particles and you suspend them in the nematic medium and you turn on the fluid, then these two particles actually attract. You can also do the same analysis with a different anchoring. So in this particular case, the nematic is perpendicular to the surface of the particle, so these hedgehogs are created. There's another interesting, the extreme limit, in which the particles now prefer to be parallel to the surface of the nematic. That's called tangential or planar anchoring. In this particular case, you also have charge separation. You have surface defects now instead of being in the bulk, and you also create systematic flows. So you can study that. Now when you put them together, pairs of particles with parallel anchoring, the first thing that happens is that they don't remain aligned. The nematic is such that they form a given angle, and that we can, for some reason, they peak around 40 degrees. This is non-experimentally. You apply the field and they actually move. There's a certain complex flow that you create, and in this particular case, it turns out that they attract. So they repel. The attraction is the perpendicular. In this particular case, the force among the particles is repulsive. I'm not going to dwell on this other than to tell you that because of talks with the folks in Barcelona, they study a slightly different realization in which they create these spherical particles with a spherical cap. So they break the symmetry not by controlling the defects necessarily in the bulk, but actually by forcing the tangential defects to be at different distances from the center by just putting spherical caps. And through same mechanisms, and I will not get into the details, but through the same mechanisms, they can actually control the motion of these particles by shining light. By shining light on the top, they can actually change the orientation of the nematic. They don't photo pattern, they do it with light, and they actually can make these particles move around and aggregate just by moving the laser. And finally, I wanted to spend 3 minutes telling you of some new experiments, not mine, but of Oleg's, which we have also modeled, in which you take a nematic liquid crystal and you submerge in it not particles but swimming bacteria. So these are active matter of sorts. These bacteria swim, and they've managed to find a class of liquid crystals that are actually not toxic to the bacteria. So the bacteria remain alive and well. Well, I don't know about well, they remain alive. It's an interesting case because you can concentrate the activity, you can increase or decrease the activity of the bacteria by increasing or decreasing the concentration of oxygen in the nematic, so you can tune their activity. At the same time, you can also change the temperature a little bit, and if you move the nematic above its nematic to isotropic transition point, then the liquid crystal becomes isotropic, and then you have an active system in an isotropic fluid, and if you lower the temperature, then you have an active system in a nematic fluid. So you can play with both parameters. These bacteria, like the case of the charges I was showing you, tend to swim preferentially parallel to the nematic. So these are bacteria with little flagella, and they move around and they follow the nematic lines. Because of the symmetry n-n, changing the sign of the nematic, because they're apolar, you have bacteria that swim in one direction and bacteria that swim in the other direction, as many in one as in the other, because there's no preferred orientation. So this is an experimental image in which they just traced out the motion of one class, the ones that move to the right, and the ones that move to the left. So this is what you would get with a +1 vortex like this. This is another +1 in which particles would aggregate and disaggregate. So you get some of them moving towards the center and others one moving towards the outside. However, there are conditions or there are cases in which the symmetry is broken, and this is one of them. If you pattern, these are again pattern configurations of the nematic. If you pattern a +1 defect, a vortex where it has a face, so it actually looks like a spiral. If you pattern a configuration like this, then the bacteria start swimming in a circle, all of them. They don't go backwards. They just, you create a systematic motion that way. You may have heard lots of talks about this. There's a lot of phenomenology for active matter, and the mechanisms are not quite known. But what's typically done in most cases is to assume that the fact that the particle is active implies that it creates a certain stress, A for active, which in this particular case would be just given by some constant, which people call lambda for some reason, and Ni and Nj are the local, N is the local director orientation. So this is the stress that's imparted on the fluid due to the active particle. The force that's the divergence of this just looks like something like this. And then the thought has been, and the suggestion has been made by those folks, that normally motion will be apolar to follow the symmetry of the underlying nematic, except when the curl of this quantity is non-zero. If this is non-zero, then you can have unipolar motion in general. This applies to active matter, but it also applies to other systems that have pusher and puller flows that are not living. Since we have a whole understanding of how this motion takes place in electrokinetic systems, we just suggested an analogy between electrokinetics and living systems, and the analogy goes a little bit like this. The density, rewritten, I had it in a different form before, and I'm ignoring now the dielectric anisotropy. The charge separation in an electrokinetic system, not in a living system, the charge in an electrokinetic system is given by the divergence of this particular product, where E is the electrostatic field, and Ni and Nj would be the local orientation of the director. So the body force, F, which is rho times the impulse field, is just given by this. So if now you have a particular design electric field, and the case we've used is just a rotating field, if you have a field that, if it's rotating, it's just the average over time gives you this particular quantity, in some cases you can actually map the electrokinetic problem into the active problem, where this would be the active stress, and this would be the standard stress that comes from the nematic. So it is possible to have an electrokinetic system in which you map the charge and the stress into a form that looks the same as the stress for an active system, but you don't have any bacteria, you just have in the cases we've done, is a rotating electric field. So one case that it's very simple to do, which I've sort of done before, is the case of two disclinations. You can have a +1/2 and a -1/2 disclination. In this case, it would be pattern, the same way they do the experiments. And if you superimpose the solutions of the -1 and +1/2, then in the electrokinetic case, and you have a rotating field, then you would have systematic flow from minus 1/2 to the 1/2. And that's what they observe in the experiments. These are traces, again, of bacteria. There are no blues. They all move from the right to the left, and they go from the minus 1/2 or 1/2 defect. So if your goal is to separate these moving bacteria or to make them move in certain directions, you could do so just by patterning disclinations in a particular cell. And then these bacteria would follow in this particular case, the flow dictated by the dipole pair that you've created. In the case of this spiral, since we have the solutions for vortices of charge +1, we can just write it down. And what you find is that the velocity fluid, the velocity of the nematic induced by, in this case, by electrokinetics for us, but in their case, the velocity of the nematic induced by the bacteria swimming, it has a very specific profile, which is R log R. I don't have the experiments, but the experiments also show R log R. So we can actually understand simply by the building blocks I discussed at the beginning what the motion of these bacteria are. There's one particular wrinkle in this case, which is that the curl of the force that we have is actually 0. So you don't need a rotational force to create rotational flow. And the reason is that as you create a stress that circles around the defect, if everything were irrotational, you would build a pressure that would be discontinuous as you go around the circle. And since the pressure cannot be discontinuous, you end up creating flow even though this curl is not 0. So this is a bit of a quick trip, but let me summarize what I try to convey to you. It is possible to have a fairly quantitative description of electrokinetic flows in a nematic matrix in which the nematic has been fixed by director configurations imposed by some lithographic treatment. This is useful it has been used already in microfluidics. It's also useful in separations and in mixing. It is possible to find these fundamental solutions that involve periodic distributions of director, elemental disclinations and their combinations, and you can use those for building blocks to design flows and particle transport. There are anisotropic effects, transverse particle mobilities, unlike the case of normal fluids. The flow has been demonstrated to be reversed, so it is understood that you can change the properties of the fluid and it will flow exactly in the opposite, which is something that's not really possible or understood in standard suspensions. Electrokinetic flows can also mimic active matter motion, and some solutions have been applied to living liquid crystals. These flows, in principle, are easier if you don't want to deal with bacteria or living matter. You can go in the lab and do experiments with an electric field, and then if you like what you get, you can try your hand with bacteria, but that's a bit easier. There's still a lot of work to do in making sure that one resolves the singularities in the director patterns and their energy and the large-scale phenomena that result from them. So there's still a bit of iffy things in the whole story I told you, but I think there's a fair understanding on the basic properties and the flow that you create in these nematic liquids and nematic suspensions. So thanks.
Thanks, Jorge. Questions, comments? Yes, sir.
I have one question. So you have made the equivalent of when you put a particle or when you put just a approachable charge. That means just a particle. So that regarding the shape of the nematic. But regarding the movement, the flow, the particle will modify the flow. The topological charge will not. Is that correct? Or is it,
so the particles move, but so the particle actually translates at some velocity that you can compute. So in the case of a particle, there's nothing being imposed on the boundaries. The defect in the matrix is just the one that's created by the particle, and they just translate as a pair at some fixed distance.
The flow of the liquid crystal through the particle, so the particle will modify the flow.
So the flow of the liquid crystal is induced by the pair particle, because the pair particle induces this charge separation that induces flow.
There is no relative movement between the...
There is relative movement between the nematic and the particle.
Yeah.
Yes. And it is true that the motion of the flow modifies the distribution of the nematic. Yes. So there is a term which comes from the viscous backflow on the nematic. Now that flow, in this case it's small. There's something called the Ericsson number. These experiments tend to be for Ericsson numbers smaller than 1. So it's fairly small. We've computed some of them, but they don't have any. But that's...
To compare to take the flow with the particle or only with the topological charge in print.
Okay.
Then the flow is different.
It's different.
And because of the particle also?
Yes. 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 So the number of rolls, I have the details, but I didn't show them. The number of rolls and the symmetry of the flow is different if you have a pair of defects, point defects, or you have a particle and a defect. Yes, that's what you asked. Yes, it's different. And it also matters if it's 3-dimensional or 2-dimensional. And the experiments are done in thin cells, but it's tricky because if the cell is too thin compared to the radius of the particle, then the defect that's elastically stable is the Saturn ring. When the thickness, the fluid is confined, so you have a particle which is not very small compared to the cell thickness, the stable defect elastically is the Saturn ring. And the Saturn ring doesn't break the symmetry in these two directions, so there's no motion. To make this move, they have to have the cell sufficiently thick so the hedgehog is elastically stable compared to the other one. But it's not 3-dimensional either, but not 2-dimensional. It's somewhere in between. Nothing happens in 2D or in 3D.
Mia.
With many experiments, there was, you have shown, I think, the electric field is AC.
Yes.
Why? I mean, why they don't? In all the experiments, I see always the alternating AC. It's not easy to put a constant one.
Yeah, so several things happen. The first one is if you put a DC field on an electrolyte, it separates in a way that screens the field, and eventually motion stops. And if you don't make it stop, then you start eating the electrodes. And so people that do experiments want to have AC fields to avoid any of this.
Yes,
Yeah, I have a question. And these topological defects travel one around the other.
Yes.
I'm thinking of these non-abelian particles, which they are function for topological.
They can.
So it depends on the path.
They can, but they have to be. You have to do something concocted, because there is for this system there is a position of smallest energy. So if you start with a different configuration, they'll relax to what's the smallest energy. But you could concoct fields that you would make them rotate. But I haven't looked at that. We haven't looked at it. But you could. If you're interested in this, there's a group in Colorado which is actually looking at not suspensions, but they're looking at funny topological defects, skyrmions in liquid crystals. But it's not related to this. In this case, they haven't made them rotate, but I think you could.
Can you just comment on this photo patterning to have the defect? Or what do you do? Is etching of this? I mean, it's not light, it's not just a shining light. What is it? No, they create a mask. And what they do with the mask is they actually modify a layer, a coating, on the inside of the wall. So they actually orient this coating. You create the mask, you shine light. When you do that, the nematic molecules, or the molecules that make the coating, like to orient themselves perpendicular to the local electric field. And that's how they create the coating on the layer. And then they put the nematic inside, and then the nematic just... So it's the same way they would force some anchoring on a regular surface, except now the surface is prepared with this photo mask. Yeah.
Any other question or comment? If not, we should thank Jorge again. He will be around tomorrow morning. If anyone is interested in talking with him, just let me know.